Justin Ray. I'm a PhD candidate in Katrina Ramanel's lab at the University of Alabama. This is my first Botany. We're recent converts from another large conference, and thanks for showing up to hear an Arabidopsis talk about a single gene. So this is really, really neat. Okay, so I want to talk a little bit today about PMRK1. That's what we're calling it. It says a receptor-like kinase in your books. We've sort of reclassified that a little bit to be a receptor-like cytoplasmic kinase, and I'll explain why. But this gene is involved in fungal defense in Arabidopsis, and it has homologs in a wide variety of species. We study how Arabidopsis defends itself against fungal pathogens, specifically powdery mildew. So powdery mildew is particularly nasty. It affects like over 9,000 species worldwide. 40% crop loss in corn, soy, wheat, rice, lots of economically and nutritionally important species. So, okay, so really quick, what I'm going to talk to you today about is I'll give you a brief overview of plant innate immunity for those of you who this is new to. I'll introduce you to PMRK1. We call it PMRK1. It stands for powdery mildew resistant kinase 1. We had to add the 1 as part of a larger family. I'll talk a little bit about where the protein is localized subcellularly and why that's important. Then what we're doing to sort of understand what PMRK1 interacts with and how the whole pathway works. And then a little bit of the new stuff. I'm working on some phylogenomics with PMRK1, and I'll talk about that too. Okay, so brief overview to plant innate immunity. So plants don't have an adaptive immune system like you or me. They are sort of naturally prepared to defend themselves against a wide variety of pathogens evolutionarily. And they do this in a few ways, and there are some important players in plant innate immunity. So there are a lot we could talk about, but I'm going to focus on sort of these 3 types of proteins today. So when a pathogen comes into contact with the plant cell, it can recognize it either extracellularly, so where the pathogen actually touches the outside of the cell wall, or it can recognize it intracellularly, say if the pathogen injects some sort of elicitor molecule into the cell. So extracellularly, you have 2 major players. You have the receptor-like kinases and the receptor-like proteins. Receptor-like cytoplasmic kinases are sort of a hybrid, and we'll talk about that on the next slide. The only difference in these 2 is receptor-like kinases have a functional kinase domain, and receptor-like proteins don't, so they just call them proteins. And then intracellularly, you have these R proteins, which stands for resistance proteins. This is a more specific form of plant defense. They can recognize pathogens even to the race level and mount sort of a system-wide response. And we'll talk about that on the next slide. Typically, when you get pathogen recognition, you get some sort of downstream phosphorylation event, so you get these cascades being activated to amplify this signal, and then you get some sort of defense gene expression. So this is a very rudimentary cartoon that I made with PowerPoint. And so imagine this is a plant cell, right? And so this is sort of like maybe what a receptor-like kinase may look like, right? So you have this extracellular domain that's able to bind some bad guy, and then you get an internal phosphorylation event going on with this kinase domain here. And then I also talked about receptor-like proteins, which are basically the same thing, but they have this wimpy intracellular domain that can't really do much. So oftentimes they have to sort of function in concert with ROKs. Intracellularly, you have these things called R proteins, which are able to bind elicitor molecules sort of in the same manner. So they actually share a lot of domains, like say leucine-rich repeats or whatever, to bind different ligands, and then they can go on to induce defense gene expression. So imagine the lightning bolts are just the elicitor molecules that are triggering all of these guys. Then you get some sort of phosphorylation event leading to a MAP kinase cascade, which is just the plant's way of amplifying that signal. A lot of times you get heterodimerization, so you get receptor-like kinases and receptor-like proteins working together to bind something, and then you get the induction of the phosphorylation event. You get things like defense response genes being turned on, you get transcription factors being turned on, and that can increase your defense response as well. And then these R proteins can just go on and directly do that. But I wanted to make mention of receptor-like cytoplasmic kinase. So they're sort of the inverse of receptor-like proteins, right? So they have the functional kinase domain, generally speaking, but they have the sort of puny extracellular domain and don't bind a lot. So by virtue of that, they work together with the receptor-like kinases a lot, right? So there's a lot of that going on, a lot of that heterodimerization, trimerization to bind different things extracellularly. So that is sort of the gist of that. So I want to introduce you to powdery mildew resistant kinase 1, which we're classifying as a receptor-like cytoplasmic kinase because it kind of does have one of those puny extracellular domains that we just talked about. So I believe the extracellular domain of PMRK1 is only 5 amino acids long, and we don't expect it to do any sort of functional ligand binding. But it is definitely doing something, and hopefully I'll prove that to you on the next slide. PMRK1, we've known about it for a while, so we identified it through microarray data, so that kind of dates it and tells you how long we've known about it, right? We're using microarrays. This was a study that showed differential expression when we exposed Arabidopsis to chitin. So chitin is the fungal elicitor molecule. So chitin is present in fungal cell walls. When a fungal pathogen comes into contact with the plant, it sloughs off chitin, and the plant is able to bind that chitin and mount some sort of response against the pathogen. We had a few questions initially. So if we knock out PMRK1, do we see different phenotypes, right? That's a pretty obvious question. Where is the protein product of PMRK1 localized? What proteins is it interacting with? Is it, period? And then more recently, I've started to ask the question, what can we learn about the molecular evolution, the evolutionary history of PMRK1? Okay. Really quickly, just to hopefully convince you that PMRK1 is important. What you're looking at, we did an infection study with powdery mildew, and what you're looking at the top here, these are just the phenotypes upon being challenged with the powdery mildew, and the genotypes of Arabidopsis, moving left to right, are the Columbia wild type here. So this is your run-of-the-mill wild type. This is your PMRK1 knockout mutant. This is your PMRK1 overexpression line. And this is a transgenic hypersusceptible line called NAG. It's basically compromised in its ability to produce salicylic acid. What you'll notice immediately is that the PMRK1 knockout is more susceptible than the wild type, but not quite as susceptible as the hypersusceptible line. And then the PMRK1 overexpression line, I mean, it looks like we haven't even touched it with the powdery mildew, right? It's super resistant, which was really interesting to us. Below that, this is just a graphical form of this, essentially. So the higher the bar, the more fungus. So we measured kinidiophores per colony, which is a measurement of sort of vigorous reproduction of the fungus. The only thing I will note about the graph below, when you start to see these differences in the white and gray bars here with the wild type and with the PMRK1 knockouts, the gray bar plants have been pre-treated, have been pre-sprayed with chitin. So what that does is you spray it about 4 hours before you actually treat it, and that wakes up the plant defense to the fungal pathogen. So you see sort of a mounted resistance in the plants that have been pre-treated with chitin. Well, what does that mean in the knockout? It means that PMRK1 is not responsible for binding chitin, right? So something else is picking up on the chitin, starting the defense response in the PMRK1 knockout. So that's why we don't believe that extracellular domain is functionally binding chitin. Okay, where is PMRK1? If it's a receptor-like cytoplasmic kinase, we expect it to be at the plasma membrane. So we tagged it with GFP. And so the top row is just PMRK1 tagged with GFP. So this is PMRK1 you're looking at here. This is your M-cherry plasma membrane marker, and this is the overlay. So yellow means it lined up perfectly, essentially, right? So see a decent amount of overlap there. In the areas where it doesn't overlap, it's more just a matter of not a lot of fluorescence in the M-cherry. The middle row here is essentially the same thing, except we treat it with salt, so that induces plasmolysis and pulls the plasma membrane away from the cell wall. So you just want to make sure that it is membrane localized and not cell wall localized. So we get pretty good overlap there with that. And then we performed a transmembrane domain deletion to see if we could sort of screw it all up in the localization anyway. And we do see some disruption of the localization. While some of it still lines up, it's kind of hard to see in this image, but what you see, and I have other images I'd be happy to share, is you see some more nuclear localization and more general cytosolic localization of PMRK1 as well. So really quick, just to summarize what I've talked about so far, PMRK1, we categorize it as a receptor-like cytoplasmic kinase. Knockout mutants are more susceptible, overexpression lines are more resistant, and PMRK1 is localized to the plasma membrane, as we expect of receptor-like cytoplasmic kinases. So that's where we're at so far. And this is sort of our proposed, this is sort of like where we think the cartoon stands now, right? So imagine, all right, so we know that chitin is actually bound by SERP1 heterodimerizing with like-5. That was shown by Gary Stacy's lab and some other people. So we don't expect PMRK1 to bind chitin, but maybe it's functioning in concert with some other unknown ROK or receptor-like protein. We don't believe it works in this sort of conglomerate of proteins, and we can talk more about that. But it's definitely doing something, so then the question is what, and how do we figure that out? And I'm doing some hybridization experiments to try to get at that now. I'm doing a cDNA library screen to assess what it may be interacting with. Then I'd like to confirm all that using yeast-2 hybrid. And then, so sort of the new stuff, I got interested in sort of the evolutionary history of PMRK1. So we're doing some comparative genomics to try to understand something about the evolutionary history of PMRK1. So I'm about to sort of change course just really quickly. So I know this is hard to see, and these are a lot of genes. So we built a gene tree of PMRK1, essentially. I'll just give you the highlights. So a lot of the bootstrap values are not that great, but it's sort of expected when you have this much sequence variation. So what you'll notice here, this is brassicasee. This is PMRK1 here, this AT5G46080. So brassicasee falls out on a longish branch. This is flax and it's out of place, so that will be probably get rid of that. So disregard that for now. But this is brassicasee and it's falling out on a relatively long branch, so maybe something interesting is going on. We found the same thing with paralogs of PMRK1 here and here. Representative of the sort of gamma triplication event for all angiosperm, or for eudicots, excuse me. We can talk more about that later. I gotta hurry, I'm sorry. And this is the third example of that triplication event. Notice Arabidopsis is not present here, so that third paralog has been lost for some reason. Could be interesting. I'm going to skip over this last slide, and I will thank you for your time and take any questions.
All right, we do have time for questions.
Hey.
Okay, so does that only, like, are brassicasee the only ones that have this gene? Is that what you were indicating, or?
No, but what the long branch to brassicasee implies is that something is happening over evolutionary time. So there's some difference about brassicasee that makes it different than the rest of the tree.
And does it only affect powdery mildew, or is it other fungal pathogens as well?
So we haven't tested other fungal pathogens besides powdery mildew. Chitin can mount a broad scale resistance to other fungal pathogens. So it's likely that it could provide some resistance to other fungal pathogens. I'm actually testing some of those R proteins that are specific to certain even races of pathogens right now. So I hope to find out more about that in the next month, actually. So we'll see. Right now,